Welcome class to a human anatomy. This is AP150 at Cerritos College. This will be an introduction chapter for the first two chapters. It's a little bit of a longer PowerPoint, and you'll notice in these lectures, they will be shortened lectures because I want to try to get you guys to watch the whole thing. So I won't be going in detail with all the slides. So you should use the resources that you have on Connect and through the online textbook to kind of supplement your studying. So my name is Elizabeth Bamer. I go by Beth. You can contact me at my email listed here. I will be holding office hours on Mondays from 7 to 8 p.m. Email me first just to verify that you'll be showing up, and then I'll provide the Zoom link for you. And I'm always happy to meet with you if you have questions about anything. All the lecture and lab PowerPoints will be posted in the modules in Canvas. Although you guys are enrolled just in the lecture section, you should have a separate Canvas page for your lab section. All right, so anatomy here, and I think I skipped here. Anatomy is the study of structure, and physiology is the study of function. So anatomy and physiology are extremely related. Microscopic anatomy looks at structures too small to be seen with the unaided eye. So we look at cytology, which are cells, and histology, which are tissues. Gross anatomy are structures that can be seen with the unaided eye. This is the structural organization of the body, beginning at atoms and starting through organization. Your textbook and Connect will take you through this structure organization, which you guys should know. These are characteristics of all living things. An overview, I'll let you read through these on your own. You should know this too. Homeostasis is any self-regulating process by which an organism tends to maintain stability while adjusting to conditions that are best for its survival. So this can be temperature, your body will sweat to release heat to cool down, or if it's cold, it will shiver, the muscles will move to try to warm up again. There's negative feedback and positive feedback types of homeostasis. Negative feedback is where the stimulus, for in this example, eating food results in rising blood glucose levels. So the high blood glucose level will be detected, and then your body will do mechanisms to try to lower your blood sugar levels. So that's negative feedback, reversing the change. And positive feedback is reinforcing the change. So for example, in terms of breastfeeding, baby feeds and continues sucking, providing that stimulus, which will tell the hypothalamus to release oxytocin, which tells the breast to eject its milk. So this is a positive feedback. More examples of negative and positive feedback are shown here. Introduction to organ systems, you have 11 organ systems in the body, and each organ system contains organs that help to perform a specific function. These are the 11 organ systems listed on the following slides that you should be familiar with, and you'll go over these in lab as well. Anatomic position is extremely important in the healthcare field to know how to describe where structures are on a patient. So first we describe anatomic position as standing upright, feet parallel, head is level and looking forward, the arms are at the side of the body, the palms are facing forward, and the thumbs are pointing away from the body. Again, this is important for describing where structures are located, and there's a variety of terminology that you'll go through in lab that'll describe where these structures are in relation to each other. Anatomic planes, this is how we divide the body into different planes. The frontal plane, the horizontal or transverse plane, the mid-sagittal plane, and the oblique plane. These are cuts in those planes on a specimen. And again, this is extremely important in the healthcare field if you're looking at an MRI, an X-ray, if you're assisting in a surgery. They'll use these words, mid-sagittal, transverse, horizontal, frontal, coronal, as they're making cuts or as you're looking at different views of an MRI. Anatomic directions are terms that describe the relative position of one body structure to another. And these directions in relationship to each other are shown here. And you will be going over this in lab, but it's extremely important to understand these anatomical directions as well. Regional anatomy, we divide the body into an axial or appendicular regions. The axial region is the head, neck, and trunk. the vertical axis of the body, and the appendicular region is all the upper and lower limbs and the associated structures with those. These are more human body regions, their name, their description, and where they're located. Body cavities, so your organs inside your body aren't just floating around and bumping into each other. They're all enclosed within what we call body cavities. And you can see the different body cavities here. Again, you'll be covering a lot of this in lab because you'll be going through activities where you'll have to place different organs in the body cavity that they're located in. So you should be familiar with what organs will go into the thoracic cavity, the mediastinum, the abdominal cavity, the pelvic cavity, and the cranial cavity. And this chart helps you with that too. Cavity membranes, ventral cavities are lined with a serous membrane, which we divide into a parietal and a visceral layer. Whenever you see the word parietal, that will line the internal surface of the body wall, and the visceral layer covers the external surface of that specific organ. Both layers produce a small amount of serous fluid to protect against friction between moving organs. So it just provides a level of protection. The thoracic cavity, the heart, is located in the middle compartment called the mediastinum. And I'll let you read through what the pericardium is and the pericardial cavity. The pleura will be a two-layered serous membrane that lines the lungs. The abdominal cavity then, or the abdominal pelvic cavity, can be divided into the abdominal and pelvic cavity, and it will also have membranes to it. And you can see here the abdominal pelvic quadrants and what organs are located in which quadrant. Cellular functions then, if we start talking about cells, these are all functions of cells. They can cover, they line, store things, move. They serve as a connection, defense, communication, and reproduction. This is a prototypical cell having a plasma membrane, the outer barrier, the cytoplasm, and then the nucleus and the control center. The cytoplasm are all materials between the plasma membrane and the nucleus, and it includes those structures listed. The cytosol is a viscous fluid containing water and many of the dissolved substances which are listed there. This goes through some of what those are. Organelles is what we'll really focus on. They're membrane bound and non-membrane bound, and they will have specific functions within the cell. These are different organelles. More of them listed there, which you should all be familiar with. And the next couple slides will take you through each organelle, which literally means tiny organ. They're membrane bound. You should be familiar with what each organelle does and what its function is and where it's located within the cell. Smooth ER is smooth due to the lack of ribosomes, unlike rough ER. And smooth ER is really important in the detoxification of drugs, alcohols, and poisons. So you'll find a lot of smooth ER in your liver. The rough ER will help with protein synthesis because it has ribosomes on it. The Golgi Apparatus, I always call the Golgi Apparatus, it's kind of the modification, packaging, and storage center. So I compare it to like an Amazon distribution center. It takes in orders, it modifies packages, sends them out, and tells them where to go. And that's what the Golgi Apparatus does, but with proteins. Lysosomes are important in the digestion and removal of waste products in the cell. Mitochondria is the powerhouse of the cell. It creates the ATP through cellular respiration. Ribosomes are comprised of two subunits that are responsible for protein synthesis. The cytoskeleton, so these are non-membrane bound. They are microfilaments, intermediate filaments, and microtubules, and they help a lot with supporting the structure of the cell, organizing the cell. They're important in cell division, and they help facilitate movement. Cilia, flagella. Flagella, the only cell in the body that has a flagella is the sperm cell, so it helps with movement. Cilia are little tiny hairs on the surface of certain cells that move objects across their surface. And this is a look at cilia, how it moves layers of mucus across the surface, for example, in the respiratory tract, and that is a flagellum attached to a sperm cell. You can read through microvilli, not capable of motion, but they're tiny projections from the plasma membrane. They are really found in your small intestine and they help increase absorption. The nucleus is the control center of all cellular activity. Nucleoli are dark staining bodies within the nucleus, and they'll be composed of the RNA, and these guys will help make the large and small subunits for the ribosomes. Then we get into DNA, chromatin, and chromosomes. DNA is the molecule containing genetic material that is inside the nucleus. DNA will wind around histone proteins to form nucleosomes. And this is the plasma membrane, called the cell membrane. It's just the border of the cell between the exterior and interior environment, and it'll serve as a selective passageway or barrier to only allow certain things into and out of the cell. Here's the composition and structure of the membrane. You should know what this is. It has a lipid bilayer, so 2 layers of lipids and proteins that either go continually through the bilayer or just are attached to the surface. Here describes what the phospholipids are and how they make up that membrane. The membrane proteins can be integral or peripheral. These are transport proteins that will serve the function of moving other materials within an organism. Protein-specific functions in the plasma membrane are listed here. They're incredibly important in transportation, connecting between cells, anchoring the cytoskeleton, so maintaining cell shape, enzyme activity, cell-to-cell recognition, and signal transduction. Transport across the plasma membrane, it's influenced by many factors which are listed here: concentration gradient, how soluble a lipid is, the molecular size, larger molecules will need help getting through the membrane, and the ionic charge. Transport across the plasma membrane can be passive or active. Passive is when materials flow down their concentration gradient by what we call diffusion. This does not require energy or ATP. And active transport are materials that move against the concentration gradient, and this will require energy from the cell. Here's a look at simple diffusion and how it will go from an area of high concentration to low concentration. And it gives several examples in the body of simple diffusion. So this is passive transport. Here's another look at diffusion. Over time, the molecules will travel through the semipermeable membrane so that over time, you'll get an equal concentration on both sides. We call that equilibrium. So this is simple diffusion, it's an example of passive transport. Factors that can affect diffusion are the concentration gradient, so how concentrated the beginning solute particles are. The temperature, higher temperature will increase diffusion, and the molecular weight and size. Osmosis is simply put the diffusion of water. So water will always flow from a region of high water concentration to low water concentration. And this is what we call osmosis. It works faster with the help of channel proteins called aquaporins. Here's osmosis showing water moving through a semipermeable membrane. So here's a beaker with water levels equal on both sides, but you can see there's a greater concentration of solute particles on the right side of the membrane. The solute particles are too big to go through the membrane, but water is, so water will travel by simple passive transport through the membrane to an area of higher solute concentration to try to get an equilibrium concentration on both sides of the membrane. How does the water know where to go? The isotonic, hypertonic, and hypotonic describe the solution in relation to the concentration of the solute particles within it. Tonicity, a hypertonic, the concentration of solute in the solution is greater than the concentration in the cell, so water will leave the cell. So if we take a look, first of all, just an isotonic solution. Iso means equal, so the concentration of these green solute particles is equal inside and outside of the cell. So water will freely move in and out of the cell, but there will be no net movement of the water. So water will maintain its normal shape. In a hypotonic solution, a cell is placed in a solution of less solute concentration. So water will move into the cell and the cell will swell. We call that lysis, if it will end up splitting. In a hypertonic solution, water will leave the cell because it'll move into a hypertonic solution where there's a greater solute concentration outside of the cell. And we saw that crenation when the cell shrivels up. And this is another look at what happens to cells, whether they're placed into a isotonic solution, a hypotonic solution, or a hypertonic solution. You should be familiar with these three types of solutions and what they mean as well. This is a great slide showing how water always will move from a hypotonic solution to a hypertonic solution. That just means that water will always move into a place where there's more solute to try to create an equilibrium and spread out those solute particles. Facilitated diffusion is also passive transport, but all it does is it uses transport proteins for larger polar molecules that need assistance for crossing the membrane. Passive transport can also be filtration, which is just the movement of both liquids and molecules across the plasma membrane. So this is filtration. It's the form of diffusion, and it's driven by the pressure of fluids or blood pressure in humans. Active transport then is the movement of a molecule against a concentration gradient. It requires ATP. One of the key examples of active transport is the sodium-potassium pump, where sodium and potassium are moved in opposite directions against their concentration gradient. Another example of active transport could be bulk transport, which is exocytosis and endocytosis. Here's the sodium-potassium pump, moving sodium and potassium against their concentration gradients. You'll learn a lot about this in physiology when you'll talk about action potentials. Here's exocytosis, is secreting something out of the cell. And endocytosis is taking something into the cell. We divide endocytosis into phagocytosis or pinocytosis. Phagocytosis means cell eating, so larger particles, and pinocytosis means cell drinking, or smaller particles are being taken into the cell. Adenosine triphosphate is the energy that every cell in your body needs. It's called adenosine triphosphate because it has 3 phosphate groups attached to an adenosine. Through a chemical reaction, when a phosphate group gets basically released, energy is put out. So energy is in the form of ATP. Cellular respiration from one glucose molecule. Cellular respiration is the way that your body takes the food it eats, such as a glucose molecule, and turns it into ATP, which is what every cell needs for energy. This is in what we call cellular respiration. And the 3 steps of cellular respiration are glycolysis, the citric acid cycle, and the electron transport chain. These last 2 steps occur within the mitochondria, and aerobic respiration occurs in when there's oxygen available, and you'll get a lot of ATP put out. Anaerobic respiration occurs without oxygen, and you'll see how the number of ATP greatly is reduced. DNA is used for protein synthesis. It carries genetic information. It's a double-stranded helical molecule. It replicates itself before it divides, and it provides instructions for making all the proteins in the body. Protein synthesis, the DNA will make a copy of itself and then be called what we call RNA, and it'll be transported out of the nucleus, and the RNA will go into ribosomes to create proteins. There's 4 nucleotides used in DNA. The bases are cytosine, guanine, adenine, and thymine. They're held together by weak hydrogen bonds. They're all complementary, so thymine and adenine will always pair up, and cytosine and guanine will always pair up. Ribonucleic acid structure, RNA comes in 3 flavors. These are the players in protein synthesis: messenger RNA, ribosomal RNA, and transfer RNA. They are made of ribose sugars and phosphates held together with covalent bonds. And instead of thymine, a thymine is replaced with uracil in RNA. Transcription is, in order to make a protein, we have to make a copy of DNA that tells us what is needed. So DNA will be transcribed into RNA. So first DNA is replicated, and then it will be transcribed into RNA, and then RNA will be translated into a protein. Translation is once we make the messenger RNA, it will now travel into the cytoplasm to make a protein. What organelle is used to make the protein? That will be the ribosome. These are different types of protein examples of what some of their functions are. Cellular division is how all cells will divide. Most cells go through cellular division called mitosis, except for your sex cells, which go through meiosis. Mitosis is a fundamental process. The cell duplicates all of its contents, its chromosomes, and splits to form two identical daughter cells. Mitosis and meiosis, you should know the differences of. They're listed here, and some similarities are shown in the middle. Enzymes, this is what enzymes are important for. They're special proteins that play an important role in helping to make chemical reactions go more smoothly or speeding them up. They speed up or catalyze a reaction while remaining completely unchanged, and that's what enzymes do. Enzyme substrate complex, an enzyme will have an active site which the substrate will bind to. That complex then at the end of the chemical reaction will produce the products. So the enzyme isn't changed at all in the reaction, it just helps speed up the chemical reactions. Factors that influence enzyme activity are listed here: the pH, the temperature, the amount of substrate, and the amount of enzyme. More functions of enzymes are shown here. And acids, bases, and buffers. An acid is a chemical compound that donates hydrogen ions, and a base accepts hydrogen ions. The pH scale describes the acidity. 7 is completely neutral. Anything over 7 is considered basic, and anything less than 7 is considered acidic. And those are examples of acidic and basic solutions. Buffers resist pH change, so these are very important when you're doing, especially in the lab, different experiments. Acidosis is a condition when the blood becomes too acidic, and this is a look at the acidosis cycle. Alkalosis is when the body has too many bases. Here's examples of functions of carbohydrates: energy, structural and stored energy, and examples of lipids. And I'm just going to go back here. Again, this takes us to the end of chapters 1 and 2. I'm trying to make these lectures a little bit shorter so that you try to listen through the whole thing, but that you also focus your attention on using the Connect resources to help you with this. Hope you guys are all doing well, and we'll see you for chapter 4.